आवाजाने दोन्ही येते. Okay, I am very happy that most of you have attended the class today. Now you are just being surprised to see that there is something going on here. It is nothing but our session is live on YouTube. It is after 2 years we are trying it. That's why exactly what is going on. If somebody reports, we don't know. Okay, it is now live stream, but it is only for those who are not the students of SRTR biochemistry, or for students of SRTR biochemistry who want to learn biochemistry afterwards. And mind way today's lecture will be restricted to this area only because it has to be seen by others. I am not sure whether my face is prettier to see by others, but we will continue with this. If you have got YouTube with you, you can just see whether this is streaming or not. Last time, what we have seen is the Gibbs free energy, that is denoted by delta G, then standard Gibbs free energy at pH 7 is denoted by delta G0 prime. I hope all of you know the YouTube channel. All of you have subscribed it. Those who are seeing it on YouTube, I request them to kindly subscribe the channel so that we can do the streaming live with the help of mobile. Okay? Now, this particular delta G, that is Gibbs free energy or standard Gibbs free energy, that is useful for the work, whatever work we do. Don't go for it. Now when I am live, don't go for the virtual. Now next thing that we are going to see today, it is around 10:45. It is quite late for starting of our lecture, but I will try to be PT, means to the point for today's lecture. Now we know that whatever energy that is captured in our body, that is in the form of a high energy phosphate bond, and it is ATP, adenosine triphosphate, for which the energy of liberation of energy is around 7.3 kilocalories per mole. So that is what free energy. Now what is the importance of ATP? We know that this is the energy currency. We already have seen that energy is, there are two laws, first and second law. If we combine together, then we can see that energy is transformed, or it is transferred from one part to another part, and second is there is increase in the randomness. Delta S, that is entropy, is raising. It is like that. Now ATP has got a special place in coupling reactions. So it takes energy from higher energy compounds like carbamyl phosphate, phosphoenol pyruvate, or from creatine phosphate. These are having more than energy of or free energy that can be given for the formation of ATP. So previously I have not shown this mark, that is negative. It indicates that this is exergonic reaction. So these reactions or these substances, they have got higher energy in the form that they can donate their energy to the ATP or for the formation of ATP. This is around minus 13 kilocalories, 11 kilocalories. Likewise, means from these substances, energy can be captured by ADP and inorganic phosphorus for the formation of ATP. And whenever ATP is there, it can give its energy to glucose 6 phosphate. This formation of glucose 6 phosphate, glucose 1 phosphate, glycerol dehyde 3 phosphate. These are having lesser energy, like 3.6, 3.2 or 3.3, something like that, or glucose 1 phosphate. It is having slightly higher energy of, this is minus. So these are exergonic reactions, but their energy level is such that they can accept energy from ATP. ATP accept energy from this. So ATP is a central, metabolically having a central position. It has got metabolically central position, means it accepts energy from higher compounds, and this can be given. So necessarily what this does, this will create a condition where these higher compounds are coupled with the lower energy containing compounds, higher energy to lower, through ATP. So it is not necessary for all the reactions to carry these higher energy compounds nearer to the lower energy compounds. It can be done with the help of ATP. So this is a metabolic importance of adenosine triphosphate. Now this adenosine triphosphate, yesterday we have seen that it is synthesized by two ways. One is substrate level phosphorylation. Now this substrate level phosphorylation, as you all know, the energy it is given at the substrate level itself. Substrate, it has got higher energy capacity. It can donate it to the ADP for the formation of ATP. And we have got three examples of this. Two in glycolysis. I hope all of you have studied this. Okay. And one in PCA. That is dicarboxylic acid cycle. So two reactions in glycolysis. One is phosphoenol pyruvate kinase, and second one is glycerophosphate kinase, bisphosphoglycerol kinase, where 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, and 1 phosphate is donated to ADP for the formation of ATP. So these are the two examples in glycolysis, and one in succinate thiokinase in PCA cycle, where GDP is converted to GTP and GTP is used for the ATP synthesis. So here there is substrate level phosphorylation. And second important thing what we are going to study today is where ATP is synthesized with the oxidation of substrate. So there occurs oxidation of substrate and ADP is converted to ATP. This is phosphorylation. That's why this particular process is called as oxidative phosphorylation. Oxidative phosphorylation is nothing but when substrates they are oxidized. What do you mean by oxidized? Like glucose is oxidized by the way of glycolysis, then it enters into the TCA cycle. This is breaking down of a substrate, glucose. Okay. There occurs oxidation, and this oxidation is coupled with the phosphorylation. That's why this particular thing is called as oxidative phosphorylation. So for the oxidative phosphorylation to occur, what we require is first oxidation. Okay. So in our body, oxidation occurs in a biological system. That particular thing means oxidation occurring in a biological system is called as biologic oxidation. And second is phosphorylation. So two things, biologic oxidation and phosphorylation, combinedly they are going hand in hand for the oxidative phosphorylation. Now mind well that all the oxidation processes in our body need not be related with oxidative phosphorylation. Okay. But most of the times oxidation occurs for the synthesis of ATP. So apart from oxidative phosphorylation, oxidation is also used for other certain things that we study later on today's session. Okay. So this is oxidative phosphorylation. Now first we will see what is oxidation. What is oxidation? In a biological system, you know what happens in a chemical system, what happens in biological system, or you can just describe what oxidation is. Yes? Anybody? Yes? Correct? It is addition of oxygen. Then it is removal of hydrogen. Then loss of this is oxidation. Addition of oxygen, removal of hydrogen, or loss of electron or removal of electron is oxidation. The reverse thing is reduction. That is removal of oxygen, addition of hydrogen, and gain in electron. Or you can say addition of electron is reduction. So most of the times in biological system or what we are going to study, this thing happens. Loss of electron or gain of electron. Loss is oxidation, gain is reduction. So in the biological system, oxidation reduction occurs with the help of addition or removal of electron. So you can remember it by the mnemonics. LEO, LEO, loss of electron oxidation, gain of electron reduction. LEO. So you can remember, this doesn't have any meaning. If you have got some meaning, then you can tell me. Most of the times we do things which necessarily do not have any meaning for them. Okay. So medical students do all the things most of the times which do not mean anything. That's why we are studying biochemistry. So this is oxidation reduction reaction. For example, here it is ferrous. If we add electron, ferric, if we add electron, it is converted into ferrous form. So this is reduction. Now ferrous, if it gives out electron, get come into ferric form, this is oxidation. So most of the times in our biological system, these are transition metals which oscillate between oxidized and reduced form, like ferrous ferric, cuprous cupric. So these are the forms of metal ions that occurs in our body, and this particular oxidation reduction, it occurs in our body. Okay. Now there are certain enzymes which are required for the biological oxidation. So there are certain enzymes which are required for biological oxidation, and these enzymes, they come from class 1 of enzyme class. You know enzyme classification. So class 1 is oxidoreductase. So this is oxidation and reduction. So the enzymes which carry out oxidation reduction reaction, they are called as oxidoreductase. This is given class 1, OTHLIL. Okay. This is the 6th class and 7th is upcoming class. So these are the enzymes, class 1 enzyme oxidoreductase. They are involved in biologic oxidation. Now what is the role of biological oxidation? We know that it is required for the energy. Now if oxidation is occurring in our body without any control, then what happens? You just tell them that lecture is going on. So what happens in a system if oxidation do not control? Means exactly what happens. What we see if we oxidize any wood, it is just burned out. So a human being or living thing don't burn out because oxidation doesn't occur at a stance in a given situation. It is not uncontrolled, means it is not just like explosion. So there are systems in our body where biological oxidation is controlled. Now if it is controlled, so whatever energy that is liberated, it is not at instance. Okay. So energy is liberated in a stepwise manner. Okay. This energy is liberated in stepwise manner. And when this energy is liberated, when oxidation reduction occurs. And oxidation reduction when occurs? Now if something is reduced, it is not in isolation. There should be something which is oxidized. So there must be two things, one get oxidized, another get reduced. For example, if A, okay, plus B. So if this particular thing is oxidant and this is reductant, means one is giving electron, another one is accepting electron, then and then only the things occur. Okay. So there should be always oxidation and reduction going on hand in hand. It is not in isolation. Whenever one thing donates electron, there should be another acceptor. So this is controlled stepwise, where oxidation reduction process occurs from donor to the acceptor. And most of the time, this particular thing, it occurs whenever needed. Okay. Now if you burn wood outside, whether it is in summer season or winter season or very cold season, okay, it will burn. It doesn't depend on your temperature of outside. But these particular things, they are isothermic, means they occur at given temperature and whenever needed. It is not like combustion. So it is as per need. This is as per need. Whenever it is needed, then and only oxidation goes on, and this occurs at body temperature. It is around 37 degrees centigrade, and internal value body pH is around 7. So biological system, it is a controlled oxidation in a stepwise manner, where electrons are transferred from donor to acceptor as per body need, at body temperature and at body pH. Okay. And what is the use of this? This is used for the synthesis of ATP most of the times, or for the maintenance of body temperature. Okay. So most of the times ATP is synthesized or energy is liberated as heat for the maintenance of body temperature. So that is a comprehensive definition of biological oxidation. So oxidation occurring in a biological system in a controlled stepwise manner where electrons are passed from donor to acceptor as per body need at body temperature and body pH, most of the times for the synthesis of ATP or maintenance of body temperature or other reactions. This is a comprehensive definition of biologic oxidation. Okay? It is simple, it is oxidation occurring in the biological system, and this is a comprehensive thing. Controlled stepwise from donor to acceptor as per need at body temperature and body pH. Okay? So, we have seen what biological oxidation is or biologic oxidation is. We have seen that it has got enzymes which comes from oxidoreductase group, and there are certain subgroups of this oxidoreductase. They are number 1, oxidase. I hope you just listen to me. I don't want to listen to you now. So, oxidase is first class of enzyme where oxidase are the enzymes which uses oxygen as hydrogen acceptor. So, how will you define oxidase? Oxidases are the enzymes which uses oxygen as hydrogen acceptor. So whenever this is the substrate which can donate hydrogen, so whenever this donates hydrogen, then requires oxygen. Okay. And what is the product? Product is hydrogen peroxide if oxygen is used, or water. So this is the role of oxidase enzyme. So it uses oxygen as hydrogen acceptor. That's why hydrogen peroxide or water is synthesized. Getting me? Now what is the example of this oxidase? The example of this oxidase is where H2 is synthesized is amino acid oxidase is the enzyme. So amino acids, they are oxidized, okay, to amino acids and hydrogen peroxide. Amino acid oxidase is the enzyme. And this requires FAD, okay, and FML. If it is L-amino oxidase, it requires FML with it. Okay. So amino acid, what is this FML? Flavin mononucleotide. Okay. Or it requires FAD. Flavin adenine dinucleotide. Okay. So these are flavoprotein containing oxidases. So final product through FMNH2 or FADH2, then they are converted to the amino acid and hydrogen peroxide is liberated. Now there are another amino acid oxidases where FMN is used or FAD is used, where instead of hydrogen peroxide, so there are two enzymes. One is L-amino oxidase and D-amino oxidase. D-amino oxidase will give water. Now we don't have D-amino acids in our body. What we have got is L-amino oxidase. So this is one example of oxidases. Now these oxidases, they are flavoprotein dependent, means they have got FMN or FAD in it. Now second example is of importance, that is called as cytochrome oxidase. That is called as cytochrome oxidase. Now name itself indicates, cyto means cell, chrome means color. So these are colored proteins which are present in cells and they contain heme or heme, whatever you spell it. So they contain heme in it. And you know what heme is, because you already know the heme protein, hemoglobin. So this contains 4 tetrapyroline and in the center iron is there in the form of ferrous. So this also contains heme, okay, and this heme, there are two types of heme are there, A and A3. They are denoted as A and A3. Okay. Now this heme, which is a part of cytochrome oxidase, the iron that is present in ferrous form, okay, it gives out its electron and can be converted into ferric form. Or whenever it accepts electron, it can be converted into ferrous form. So the iron in the cytochrome oxidase heme, it oscillates between ferrous and ferric form. This is the difference between hemoglobin and cytochromes. Cytochromes, they are used for oxidation reaction to occur, and if what is the form of iron that is present in hemoglobin? It is ferrous. If it becomes ferric, then that is called as methemoglobin, and methemoglobin is no longer able to bind with the oxygen. So the function is lost in case of hemoglobin, whereas it is a prerequisite for cytochromes to oscillate between ferrous and ferric form. It is stringent for hemoglobin, it is always ferrous, whereas here it oscillates depending whether it is oxidized or reduced in the ferrous and ferric form. So this heme, there are two types of heme A and heme A3. Previously it was just called as cytochrome oxidase or heme A3, but later on we come to know that it is AA3, and this particular cytochrome is called as cytochrome AA3. And this is the final thing that occurs in the biological oxidation in the mitochondria. We will see later on in the details. This is the last component of electron transport chain. What electron transport chain we will again see. So this is last component of electron transport chain, where electrons from electron transport chain, hydrogen from matrix and oxygen, they combine together and forms water. So this particular reaction at the end of electron transport chain, it occurs with the help of cytochrome oxidase. So cytochrome oxidase is a last component of electron transport chain. Okay? So this is first type or first component of enzymes that we have studied. First is oxidoreductase is the class from which we have seen first oxidase. So cytochrome oxidase and other amino acid oxidase. We have seen amino acid oxidase, they are present or they have got FAD, while cytochrome oxidase it is having cytochrome A3 or it contains heme A3 and it is the last component of electron transport chain. So this is first. What is the second class of enzyme? These are described as dehydrogenesis. As name indicates, these dehydrogenesis, they utilizes hydrogen removal from their substrates and hydrogen is added to the other substrate, so that one substrate is reduced and another one is oxidized. So whenever one substrate it donates hydrogen, it get oxidized. So dehydrogenase, what it does? This is simple thing that occurs. This is simple thing that is occurring over there. ठीक है? Just removal of one or removal of hydrogen. So one substrate it gives hydrogen, another accepts. Now these dehydrogenases, there are different types of dehydrogenases. One is NAD dependent dehydrogenases. Nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. This is one type of coenzyme that is required for the enzyme dehydrogenases. Now this coenzyme it accepts one proton from substrates. So what are the examples? You probably know the examples of glycolysis and TCA cycle where dehydrogenases they are used, where suppose this is the substrate and NAD is there. This is converted into A plus NADH plus H plus. So NADH it accepts hydride from the substrate. Hydride means hydrogen plus one extra hydrogen plus electron, it forms a hydride. So it has got one extra electron and remaining proton or remaining hydrogen is liberated in the medium as a proton. So all the reactions, whether it is pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, for that example glutamate dehydrogenase that you are using for the estimation of urea, where NADH is formed or NADH is consumed. So NADH is formed and H plus is liberated in the medium. So these are NAD dependent enzymes. So enzymes of all glycolytic enzymes which are dehydrogenase enzyme. Pyruvate dehydrogenase is there. Then in TCA cycle, alpha ketoglutarate dehydrogenase, succinate dehydrogenase. So there are lot of dehydrogenase enzymes are there which are NAD linked, means which uses NAD as acceptor of reducing equivalents. Now we are talking about electron, okay, then proton or hydride. Combinedly, these are called as reducing equivalents. Rather than telling each time it is electron or proton or hydride, we call it as reducing equivalents. Okay, it may be anything out of this. So reducing equivalents, they are accepted or they are transported from the substrate to NAD. NAD is converted to NADH and H plus is free in the medium. So examples we have seen in glycolysis and in PC cycle, for that matter, for when we study fatty acid oxidation, in case of beta oxidation, there are different dehydrogenesis in beta oxidation also. So they are NAD dependent, and there are certain which are dependent on FAD. So this is one dehydrogenase, that is NAD dependent. Another dehydrogenase is flavoprotein dependent, FML dependent or FAD dependent. Dependent means which require this thing as coenzyme for enzymatic activity. Now you know FAD is used as a coenzyme in various reactions. We have already seen one. Now in case of beta oxidation, first step of fatty acid oxidation, it depends on FAD. FAD is converted to FADH2 with the help of flavoproteins. Acyl-CoA dehydrogenase is the name of enzyme. Acyl-CoA dehydrogenase, which utilizes FAD as its coenzyme, and FAD, when it accepts the reducing equivalents from substrates, it is converted into FADH2. It is converted into FADH2. Okay. So this is how the conversion occurs. Now what remains is FML. What remains is FML. This is important as far as electron transport chain is concerned, because the first component of electron transport chain is a complex which has got enzyme called as NAD dehydrogenase. See name, or NADH dehydrogenase. This is enzyme, okay, but this enzyme, it requires FML. Enzyme is NADH dehydrogenase, but it requires FML, and this is the component of first complex of electron transport chain. When we study electron transport chain, this will again come. So here NADH, that is formed, it gives proton or reducing equivalents to the FML, and FML is converted into FMLH2. Of course, it requires another H. So NADH plus H plus with the help of NAD dehydrogenase, converts FMN to FMNH2. So reduced flavin mononucleotide. This is nicotinamide adenine dinucleotide reduced. When H is added, this is called as reduced nicotinamide adenine dinucleotide, or when it is NAD, it is called as nicotinamide adenine dinucleotide. Reduced nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide. Reduced flavin mononucleotide, flavin mononucleotide. So instead of calling it as FMNH2, we call it as reduced flavin mononucleotide. So it is rather difficult all the time to complete reduced nicotinamide adenine dinucleotide. So it is better, or it is in practice, that we call it as NADH. But don't say NADH2. It is not correct. It is always NADH, and it is always FADH2 or FMNH2. Got it? So this is another enzyme, that is FML dependent. So we have seen enzyme which is dependent on NAD, we have seen enzyme which is dependent on FAD, and we have seen enzymes which are dependent on FAD. Now what is the role of all these dehydrogenases? So what they do, they directly collect the reducing equivalents for the substrate, like from glycolysis, from TCA cycle, from breakdown of fatty acid in the form of beta oxidation. All these reactions, they occur inside the mitochondria, in the mitochondrial matrix. So whatever energy that is stored in the substrate, or higher energy level, that is captured with the help of reducing equivalents, and these reducing equivalents, they are then transported to the, or they come into the contact with the electron transport chain, where electrons, they are snatched from these NADH or FMNH2, and then they are transported through electron transport chain, and this occurs in a stepwise manner, where energy is liberated, and that energy is used for the capture of or for the synthesis of ATP. The liberated energy is captured for the synthesis of ATP, and this whole process is phosphorylation, which is coupled with the oxidation. So why we are studying this? We are studying this because to know how exactly our body utilizes whatever we eat in our diet, how these substrates, they are oxidized, why they are oxidized, and if they are oxidized, what happens, and how energy is captured. So energy is not only liberated, it is captured and then utilized for the another useful works. Whenever required, ATP is used for that particular purpose. Okay, so these are certain things that we must know. So this NAD dehydrogenase and FAD dehydrogenase, they play important role in metabolism. Now metabolism is made up of two things. One is anabolism, another one is catabolism. Anabolism is just synthesis which requires energy. It is synthesis of substrate, and catabolism is breakdown, and breakdown occurs with the help of or in the process of oxidation. So whenever substrates are broken down, that is called as catabolism, and both combinedly, anabolism and catabolism, is called as metabolism. So catabolism, for that to be specific, is used for this purpose. It is used for this purpose, catabolism, so that the substrates, they are oxidized, and the electrons that are being transported, or the hydrogen that is liberated, they are finally used for driving the electrons in a stepwise manner through electron transport chain, that we will see in next lecture, so as to capture the liberated energy in the form of high energy phosphate bond ATP. Got it? Now there are certain other cytochromes in our body. To ask them that YouTube is going on. Second is dehydrogenase, that we have studied. Before going to third, there are cytochromes in our body. which we have already seen that they oscillate in between ferrous and ferric form. So, if we take one example of it gives one electron. Okay, this electron used by another cytochrome B. This is arbitrary examples. We will see detail when we study electron transport chain. So this is taken up by another ferric form is converted into ferrous, okay, by giving one electron. Then what happens here? One electron is given, that is accepted by another ferrous or ferric, that is converted into this is ferrous. It gives one electron, get converted to ferric form, and whenever one electron is given, that is accepted by another ferric form, it is converted into ferrous form. This ferrous form again gives one electron, that is accepted by cytochrome C. This is A, B, C are example. This is not cytochrome A, cytochrome B or cytochrome C. This is just a numerical order A, B, chronological order A, B, C. Okay. So this will give one electron, another ferric form is converted into ferrous form. Likewise, this goes on. So cytochromes, which oscillates in the ferric ferrous form by donating or accepting electron, these are coupled reaction. See, when one cytochrome is oxidized, another is reduced. Reduction, oxidation, reduction. So one cytochrome, this is A, it is oxidized, another B is reduced. So these cytochromes, they just work like that of dehydrogenesis. That's why cytochromes can be said as the dehydrogenesis. They can be classified under dehydrogenesis. Okay. Then third one is hydroperoxidases. Oxidases and dehydrogenesis. These are the two enzymes which are involved or which are required for the oxidative phosphorylation process. Okay. Enzymes from dehydrogenesis and oxidases, they are involved in the electron transport chain for the oxidative phosphorylation. Now these hydroperoxidases, as name indicates, okay, they are the enzymes which scavenges the hydrogen peroxide or reactive oxygen species. They are classified into peroxidase and catalase. Catalase is heme containing enzyme. Now what this peroxidase does? Hydrogen peroxide plus is converted into whatever. So this hydrogen peroxide is converted into water. Now what is hydrogen peroxide? This hydrogen peroxide, where it is synthesized? Which enzyme synthesizes hydrogen peroxide? Anybody? First enzyme we have already seen, oxidases. Oxidases, they synthesize hydrogen peroxide. So necessarily hydrogen peroxide which is synthesized. Now this hydrogen peroxide is a deleterious substance, molecule, hydrogen peroxide. It can carry out cell damage by oxidation of various biomolecules. Cell membrane itself can be damaged with the help of H2O, and if the cell membrane is of RBCs they are lysed. There occurs hemolysis. So to scavenge the hydrogen peroxide in the RBC, there is another substance that is used, that is called as glutathione instead of any other substance. You know, this is glutathione is thiol containing substrate. This is biologically important peptide. This is tripeptide. Okay. Now this glutathione, it scavenges hydrogen peroxide. That's why this particular enzyme is called as glutathione peroxidase, and this is important for RBC. Okay, glutathione, instead of this you can write like this 2GSH. Okay, and this is converted into again GSSG plus H2O. This is glutathione peroxidase. This is not seen. Is it seen? Whatever. So glutathione peroxidase. So H2O2 plus plus 2GSH. This is glutathione, reduced glutathione. So this reduced glutathione is converted into oxidized glutathione and water. And here glutathione peroxidase is the enzyme. And this is important for the RBC. RBC has got glutathione peroxidase in it, and it prevents hemolysis or destruction of RBCs due to hydrogen peroxide. And we also use horseradish peroxidase. Where we use horseradish peroxidase, or we used it for the estimation of glucose. Okay. Remember, glucose oxidase peroxidase. This peroxidase enzyme, the source is horseradish. Okay. So glucose is oxidized. What is product? Same. Remember the name, oxidase. Glucose oxidase, it forms hydrogen peroxide, and that hydrogen peroxide is cleaved with the help of the substrate in glucose. Hydrogen peroxide is cleaved with the help of peroxidase, that is horseradish peroxidase. Here it is glutathione peroxide. This occurs in the cell, okay, in RBC, so that hydrogen peroxide that is being formed, that is cleaved. Okay. So this is the importance of this hydroperoxidase. Another enzyme is catalase. Now, what is the role of catalase? It is heat containing. It uses hydrogen peroxide itself as electron acceptor and electron donor to form water and oxygen is equal. This is catalase, a heme containing protein. And this action of catalase is so quick. Okay. So if enzyme is not there, and in presence of enzyme, it is 1000 times more quicker. So that is the importance of catalase, again to scavenge the hydrogen peroxide. And then there are other enzymes. They are called as superoxide dismutase. This superoxide dismutase, as name indicates, it dismutate or scavenge the superoxide radical, that is oxygen with one electron more. And this is highly reactive substance. So active that whatever comes in its way, it is destroyed. It is destroyed. Like the warriors that we have seen in dynasty of Maharashtra. So there were two warriors, you know, who just destroyed their enemies, and they never got defeated. They always won. There are two examples, two great examples that we should know. One is Chhatrapati Sambhaji Maharaj, and second one is Thorle Bajirao. So both of them, they have battled. Okay, every battle they won. So same thing. Okay, so you must remember that the radicals that have been formed, so they just destroy the things, whatever things come in their way, they just destroy. So that is scavenged with the help of enzyme superoxide dismutase. It is copper or manganese containing enzyme, or some enzymes of cell wall, they are ferrous containing superoxide dismutase. Again they convert this particular superoxide in less deleterious substances, okay, hydrogen peroxide and oxygen. So that is role of superoxide dismutase. So that is third class of enzyme. That is not third class enzyme. Third class of enzyme that we have seen in the oxidative phase. Fourth subclass. Fourth subclass of enzyme that is important for the oxidation reduction is oxygenesis. Is oxygenesis. There are two oxygenases. One which incorporates both molecules, that is O2, into the substrate, that is called as dioxygenesis. Simple. Homogenetic oxidase or tryptophan pyrrolase are the examples of this dioxygenase. Both the molecules of oxygen, they are incorporated in the substrate. Second one is monooxygenase. As name indicates, monooxygenase. So they incorporate only one molecule of oxygen into the substrate. Another one is converted into, or it is reduced to water. Okay. So this is monooxygenase. This is also called as hydroxylase, or this is also called as mixed function oxidase. So you don't remember oxidase now, because we know what oxidase is. So you simply remember monooxygenase or hydroxylase. As name indicate, it hydroxylates the substance. This is simple reaction that occurs. And what is the importance of this reaction? There occurs hydroxylation, and this is important for steroid hormone synthesis, dehydrogenesis. Okay. Hydroxylation reaction. And this is also important for the removal of foreign substances, drugs. So drug metabolism, it occurs with the help of hydroxylation. And this hydroxylation is done with the help of a system which hydroxylates, and this system is called as cytochrome P450 system. We will see later on when we study xenobiotics regarding cytochrome P450. Okay, this is present in virtually in all cells, in liver, adrenal, where dehydrogenesis or hormones or lipid synthesis, wherever hydroxylation is required, this particular cytochrome P450 takes part in the hydroxylation reaction. There are again hydroxylation reactions that occurs in microsomes, or there are certain hydroxylation reactions that occurs in mitochondria. Some of them they directly dependent on, there are again three classes of cytochrome P450. One is dependent on NAD, another one is dependent on FAD, and last one newly come that depends on B5. So these are different. You don't remember this. Just remember that cytochrome P450 is a system of enzyme which is required for the hydroxidation, and this is important for xenobiotics. Whatever drugs that we take, that is being metabolized by this xenobiotic process, means biotransformation. Drugs, they are hydroxylated, then become more polar, and then they are excreted out of your body. So that is the function of oxygenesis. Dehydrogenesis, monooxygenesis. So for biological oxidation, the first class of enzyme, oxidative, it takes part, and from this we have seen oxidases, dehydrogenesis, hydroperoxidases, and oxygenesis. These are the four classes which are useful for biological oxidation, out of which these two, they are directly involved in an oxidative process where the end product is phosphorylation, or where the end product is capture of energy. These are oxidases and dehydrogenase. While hydroperoxidases and oxygenesis, they are important for synthesis of lipids or scavenging the free radicals or conversion of various xenobiotics, means outside substances to be excreted from our body. So that is work of these two enzymes. So now is it clear that all the oxidation that occurs in our body, or biological oxidation, is not totally for the capture of energy. Some of the oxidative processes, they are used for hydroperoxidase and oxygenesis reactions, which are also important for our body. Nevertheless, we will see the enzymes which are important for oxidative phosphorylation, because we are now studying biological oxidation in context to the oxidative phosphorylation. And we have seen enzymes that are required for the oxidative phosphorylation in the form of oxidases and dehydrogenase. Now, with this backdrop of free energy chain, that is gives free energy, and the biological oxidation. So these are the two things that are mainly form the basic or basis on which the second thing, that is oxidative phosphorylation, it builds. Now the oxidative phosphorylation, it occurs in the mitochondria. For that matter, you should know what is mitochondria, what is its structure, how the electrons, or how these oxidative processes they are linked to electron transport chain that is present on the inner mitochondrial membrane for the capture of energy in the process called as oxidative phosphorylation. For the want of time, we will see this in next lecture. Okay? Any difficulty? Any problem? Is there any problem? Okay.